Good day to everyone. Thanks for joining this webinar. It is my pleasure to present here. You have already seen the plan of the talk. I will start with a co-localized AFM and classical diffraction limited optical techniques such as confocal Raman, fluorescence microscopy, and lifetime imaging. And then I will move on to the optical techniques beyond the diffraction limit, such as tip-enhanced Raman scattering and tip-enhanced fluorescence. And I will finalize the talk with what is new in NT-MDT technology in scanning near-field optical microscopy. I would like to thank the team of our application scientists. You can see the list of the people here, some of the data which you will see today, and some of the methods were generated by our applications team, and all other data I will demonstrate will come from our customers, and in the corresponding slides you will see the references to the groups and to the publications. So let me start with a few facts about NT-MDT. The company started to design and produce atomic force microscopes more than 24 years ago. More than 4,000 devices have been installed since that time. The company has 5 major offices worldwide in Russia, Ireland, Netherlands, US, and China. Each office has a team of application scientists, service engineers, and sales people to support our customers locally. And the company has more than 30 distributors worldwide and occupies about 16% of AFM world market. When it comes to AFM integration with light, that is with confocal Raman, fluorescence, tip-enhanced Raman, and scanning near-field optical microscopy, this has started at NT-MDT in 1999, and since that time a few hundred installations were performed worldwide, both in academia and industry. And now in 2013 we released what we call the fourth generation of AFM-Raman instrumentation: new electronics, new hardware, software, and new measuring techniques. More about NT-MDT AFM with optics technology can be found on this website. So here you can see probably the most universal way to integrate AFM with optics. In the middle there is an AFM probe, and on the top, on the bottom, and on the side there are objectives: 100x, high resolution, high numerical aperture objectives that each of them can be used to focus the excitation laser to the very end of the AFM tip and to collect the resulting optical signal from the tip and from the sample. To align this setup, there are multiple piezo-controlled mirrors and objectives. Every piezo is closed loop control with nanometer precision and accuracy. Different probes, of course, can be supported: standard AFM cantilevers, SNOM cantilevers, STM tips, or just sharp metal wires or quartz fibers attached to tuning forks. So basically all standard and non-standard AFM probes can be installed into this instrument integrated with optics. Different optical techniques are supported. First of all, this is connection to confocal Raman and fluorescence microscope. The optical signal can be detected by photon counting, PMT, and APD. Lock-in detection of optical signal at various frequencies is possible with the electronics, which contains multiple built-in lock-ins. Gated photon counting, time-resolved fluorescence, lifetime imaging, and the system can work also with laser confocal microscopy. The wavelength range, the standard, is visible. Upgrade options to UV and near infrared are available, and finally we have customized solutions to enter mid and long infrared light onto the end of the AFM tip. The experiments can be done under controlled environment, temperature, humidity, gases, or even in liquid, in electrochemical environment, and some experiments can also be done at external magnetic field. So this is a photo from a real installation of a system which contains objective from top, from bottom, and optionally from the side. All of them can be used for focusing laser light to the AFM probe and collecting the resulting optical signal. We call such configuration a 4-pi configuration because of the universal optical access to the tip from all sides. Of course, not every application requires simultaneous optical access to the tip from top, bottom, and from the side. That's why in most of the cases we divide the system into separate configurations. The inverted configuration, when the objective is focusing to the AFM probe from the bottom. This configuration is installed onto professional inverted optical microscope, and the upright configuration with objectives from the top and from the side. Both of these configurations can be coupled to one and the same Raman instrument, and it's easy to switch in between these two configurations in AFM-Raman and TERS measurements. This is one of the latest instruments in our AFM-Raman family. This is a fully automated AFM-Raman that features integration with a professional upright optical microscope. The microscope turret can be freely rotated with AFM engaged to the sample. If the AFM is not required, then the tip can automatically go away and later return back exactly to the same working position. So in this way, the optical microscope can be operated completely independent from AFM, or it can be operated together with AFM. Fully automated AFM-Raman and tip-enhanced Raman imaging is done with this instrument. So this is how we do co-localized AFM-Raman imaging. This is example of a graphene flake, AFM probe, and laser spot is focused in a diffraction limited spot exactly to the end of the AFM tip. While we're scanning the sample, we collect optical signal, Raman, fluorescence, and elastically scattered signal by a confocal Raman spectrometer connected to the same objective, and simultaneously we obtain AFM images by AFM cantilever by AFM electronics. So this is simultaneous co-localized AFM and Raman imaging done exactly from the same sample area. And this is a flash animation done after a real experiment. For about 1 minute we collect 100 by 100 points Raman map simultaneously with AFM topography and AFM phase. So these are different, this is a relay map, G-band intensity, full spectral Raman map where we record the complete Raman spectrum at each point, and AFM topography and phase pictures. Here is the data we generated during that experiment. That was done already a few years back, but it still attracts a lot of attention when we show it. That's why I decided to include it into this presentation. This is graphene on a gold surface. It's a monolayer flake, double layer flake, and this is multiple flake area. This is AFM topography from which we can extract preliminary number of graphene flakes in the sample, and this is Raman map, G-band intensity, and 2D band position, which directly confirms number of layers in the graphene. Lateral force microscopy gives us semi-quantitative information about friction coefficient between graphene and cantilever, and we could see that friction coefficient drops down when we increase number of layers. Electrostatic force microscopy gives charge distribution. Force modulation microscopy, elastic modulus of the sample. Kelvin probe microscopy gives us the surface potential. We were able to extract the quantitative information about dependence of graphene surface potential on the number of layers. This is co-localized AFM confocal Raman and confocal fluorescence maps of molybdenum selenide flakes. Topography shows the monolayer, bilayer, and multilayer flakes, and some contamination around the sample coming from the sample preparation procedure. On the surface potential map, this contamination on the surface is not observed, but we can see a very nice contrast between the flake and the substrate. And the flake also shows some non-uniformity of the surface potential, presumably due to contamination. This is a Raman map. Monolayer, bilayer, and multilayer shows slightly different Raman signatures. And finally, micro photoluminescence map is very sensitive to the number of flakes, because monolayer of molybdenum selenide is a direct band gap semiconductor, so we can see a strong direct exciton emission. Multilayer and bulk molybdenum selenide has indirect band gap and virtually no photoluminescence, and the bilayer is somewhere in between. It still has a photoluminescence in near infrared region, but it's much smaller than for the monolayer. Here is co-localized AFM-Raman map of mixture of cadmium sulfide and polyaniline nanowires. On the AFM topography, we can hardly distinguish between polymer and semiconductor nanowires, while on the simultaneously measured Raman maps there is a very nice chemical contrast, shows us which nanowire is polyaniline and which nanowire is cadmium sulfide. This is simultaneously measured co-localized AFM-Raman data of boron nitride nanotubes. On the AFM topography we can see nicely the structure of the nanotubes with a very high resolution. On the Raman map of the boron nitride we can see the distribution of the nanotubes on the surface, and we can also see some carbon inclusions by the graphitic G band, and these inclusions presumably come from carbon crucible used for the sample preparation, for the sample growth. Here is our recently introduced new measuring mode into NT-MDT instrument. We call it a hybrid mode. The tip moves up and down about 2000 times per second, and at each approach and retract cycle the force distance curve is recorded. That is deflection of the cantilever determining the force versus cantilever to sample distance, and online analysis of these force distance curves or post-processing of these curves, because they can be pre-recorded during the measurement. So this analysis allows to extract quantitative information or semi-quantitative information about sample elastic modulus and adhesion to the cantilever. And we are also, together with the deflection of the cantilever, with the force, we are able to measure electrical properties of the sample by measuring the current or the voltage between the cantilever and the sample, and we are also able to measure optical signals, but that will be described in details in my further slides of the presentation. This is how the hybrid mode works together with Raman. On the right hand side you can see the Raman map, the 2D band map of the graphene monolayer on a gold substrate. So from the Raman we can see that this is actually from the shape of the Raman band, and from the intensity we can judge that this is a single monolayer graphene. We can see in this circle some non-uniform distribution of the Raman intensity. We don't know whether it comes from the graphene defects or from some other reasons. If we use the standard AFM microscopy, this is the left slide, this is the phase image of the standard intermittent contact AFM imaging regime. We can see also some non-uniform distribution on the surface of the graphene, and we don't know whether this is some impurity on the surface or this is the graphene defects themselves. We don't know that, but in a hybrid mode we surprisingly can see a completely uniform image. This is actually the stiffness measured by the AFM by approaching and retracting to the sample 2000 times per second and measuring the force distance curves, and we can see the uniform distribution of the stiffness, and this is due to the fact that actually this graphene was covered by a thick organic contamination layer, and actually the classical intermittent AFM regime is just measuring this layer, the surface of the layer, while in the hybrid mode we are able to set the force, the set point force between the cantilever and the sample to high values, and we are able to pinch with a sharp probe through the contamination on the surface of the graphene and reach the graphene layer itself, and prove that the graphene layer itself has no defects and it's uniform. This is multiple mode imaging of a polymer blend deposited on the glass substrate. On the topography we can see the droplet, and we can see some signature of one type of the polymer and the matrix of a different polymer. Adhesion and stiffness maps produce very nice separation of two types of the polymer in the droplet: DC-DZ, the electric permittivity sensitive mapping, and DC-DV maps single pass electrical AFM modes, allow to extract some information about sample electrical properties. And finally, Raman map provides direct insight into the sample chemical composition. All these measurements are done from the same place on the sample. Some are done simultaneously, and a few others are done in successive scans without changing the cantilever. if we are trying to measure with a classical two-dimensional scanning Raman the very rough surface, a few nanometer roughness, we are going to see the artifacts that are connected to the fact that the sample does not stay in focus of the laser spot. While we are moving the sample, it gets in and out of focus, and the resulting Raman maps are not actual indication of chemical distribution on the surface, but the artifacts from focusing and defocusing. But when we have the AFM tip inside the laser spot, the situation is different, because the AFM feedback moves the sample up and down in Z direction in order to keep the sample always in contact with the cantilever. Thus, when the laser spot is focused exactly to the end of the tip, we guarantee, by having the cantilever and by having the AFM feedback on, we guarantee to stay always in focus with the laser on the sample, whatever the sample roughness is, and that allows to measure by Raman chemical composition of very rough samples. Here is a cross-section of thin film of kappa gallium selenium of different stoichiometry. On the AFM topography, we can see the cross-section has a very large roughness with a grain size more than 6, grain height more than 6 micrometers, and it's not possible to do classical 2D Raman mapping, but with the AFM focus track applied, it's possible to do Raman compositional mapping, and it's possible to distinguish between different phases in this thin film. This is a photovoltaic cell prototype, microcrystalline silicon islands in an amorphous silicon sample, and the AFM conductive AFM cantilever is locally illuminated by a blue light, and here is the co-localized AFM topography, photoconductivity, and Raman measurements of the sample, and microcrystalline silicon islands are perfectly resolved both in Raman, in topography, and most importantly in photoconductivity measurements. This is the example of co-localized AFM topography and spinning disk confocal measurements of a labeled cell. It's possible to do such measurements both on dry samples and cells in aqueous environments. And this is the photo of the system. This is Andor spinning disk confocal, inverted optical microscope, and atomic force microscope on the top. It's possible to overlay these images and analyze them simultaneously in the same software. This is co-localized AFM laser confocal fluorescence and fluorescence lifetime imaging of a diatom cell. The cell was locally modified by high laser pulse, and in this point of the modification we can see a different fluorescence and different lifetime imaging. Fluorescence lifetime imaging is the way to look at the structural composition of biological objects without labeling, and NT-MDT is supplying integrated AFM film microscopes for biological and for other types of investigations. So all the data I showed previously today, the optical maps in the co-localized AFM and optical imaging, the optical maps were diffraction limited. That is about 200 to 400 nanometer confocal Raman confocal fluorescence resolution. This is the diffraction limit. This is close to half of the wavelength, and if we want to go beyond the diffraction limit and to get the optical mapping, mainly Raman and fluorescence maps of the sample with resolution down to 10 nanometers, we need to apply so-called tip-enhanced Raman sketching and tip-enhanced fluorescence or tip-assisted optical techniques. In this technology, there is a nano antenna, usually metal or metallized AFM or STM probe, and at the end of this probe, if it's properly prepared, there can be a localized surface plasmon oscillations of negative electrons with respect to the positive metal core. And if the energy of this localized surface plasmon is close to the energy of the incoming laser, then it can be realized the effective energy transfer from the laser into the localized surface plasmon, and then the localized surface plasmon further emits light into the far field of the same energy coherent light, but this emission is now localized about 10 nanometers around the tip. So this is the very basic explanation of tip-enhanced optical techniques, tip-enhanced Raman and fluorescence technology. As I have mentioned already, with NT-MDT instrument it is possible to do tip-assisted optical techniques in various configurations, in inverted, upright, side illumination configuration, and the 4-pi full optical access configuration. And our customers using NT-MDT instrument at these various configurations successfully generate tip-enhanced Raman data. This is a typical tip-enhanced Raman scattering experiment. Let's take nanotube as an object. First we have to take an objective from the top, from the bottom, or from the side and focus the laser into the diffraction limited spot on the sample. With the top illumination option, that's about 400 nanometer diameter laser spot. Next thing is to take nano antenna, especially prepared AFM or STM probe that has a localized surface plasmon at the end, and to place this nano antenna on the top of the sample, on the top of the nanotube. Now we have to position the laser spot with respect to the tip to find the end of the tip. Because the laser spot is diffraction limited, this alignment can be pretty challenging, and to do that we do automated scanning of the laser spot across the tip area with a special closed loop control piezo mirror, and while we're scanning the laser spot we're collecting elastically scattered light and the Raman response from the sample. And if there is a localized surface plasmon at the end of the tip, we are going to see the peculiarities in the elastically scattered laser light intensity due to interaction with surface plasmons, and we are also going to see the enhancement of Raman signal at the place where the tip is touching the nanotube. This is the way to do automated alignment of the laser spot with respect to the nano antenna, with respect to the hot spot end of the tip. And this alignment in some experiments has to be done with a 10 nanometer scale precision and accuracy in order to receive reliable and reproducible results. That's why it has to be done in such automated regime, and it cannot be done properly by just a simple manual alignment. So the next thing after the laser spot and tip are aligned with respect to each other is to scan with the sample. This is what is happening when we are scanning with the sample. First we can see the large diffraction limited laser spot, and we can see some light localized at the very end of the nano antenna, and that light is greatly enhanced. So when diffraction limited laser spot passes through the sample, we see a small Raman or fluorescence response from the sample nanotube in this case. When the end of the tip passes through the sample, due to localization of light at the end of the nano antenna, there is going to be a greatly enhanced response, optical response from the sample, and this response is specially localized about 10 nanometers at the end of the tip. So as a result of such scanning, if we collect Raman response from the sample, we're going to get image like that. This white image, low intensity image, is a diffraction limited Raman, micro-Raman, a classical 5-field Raman image of the nanotube, and it's diffraction limited. Its width is going to be about 200, 300 or 400 nanometers depending on the objective. Then on the top of it we are going to see much higher intensity and much higher resolution Raman map of the nanotube, and this Raman map is already produced by laser light, localized and greatly enhanced at the end of the nano antenna. So this is the way how we obtain tip enhanced Raman maps, and now let's look at the real data. So now I'm going to show you a video of a real experiment, which is tip enhanced Nano-Raman, and it's going to be very fast Raman, very sensitive and very high resolution. The sample is thin organic layer of molecules on a gold substrate. Here you can see the measurement parameters, and you will see the enhancement more than 100x, 4-minute time for the whole Raman mapping, and spatial resolution less than 20 nanometers. Now let's see it. First we start with the tip. This is the tip, specially prepared tip attached to the tuning fork. This is the end of the tip. Now this is the sample itself. Now we switch on the laser. We switch on the feedback, and after we switch on the feedback, the laser is focused on the sample. We manually move the laser around the tip just to do a rough alignment, and then the next step is, as in my previous video, will be to scan with a laser spot around the area of the tip. So the end of the tip is somewhere around here, and while we're scanning, we're collecting Raman response from the sample. So here is the hot spot. Here is the place where the tip has a localized surface plasmon, and it is touching the sample, and the sample produces enhanced Raman signal due to the presence of the tip. So this is Raman spectrum away from the hot spot, virtually nothing, and this is Raman spectrum at the hotspot. The overall enhancement, the net enhancement, is more than 100. So the next step after the laser spot alignment, and the alignment actually takes only 40 seconds, as you have seen. So the next step is just to scan with the sample, and these are scanning parameters, 1 by 1 micron, 90 pixels, 30 milliseconds per pixel, and the overall Raman tip enhanced Raman mapping time will be about 4.5 minutes. So here you can see the data. It's a little bit shortened, so overall time would be 4.5 minutes, as I said. Here is the clock, and you can see it's 1 by 1 micron scale, and it's non-uniform distribution of the molecules on the substrate, and the cross-section, which we will analyze later, will demonstrate about 20 nanometer lateral resolution of these Raman maps. So again, it is very fast, it is very sensitive, because these are organic molecules and they produce very, very small Raman signal without enhancement of the tip, and this is extremely high resolution. Now, can it be done faster? Can it be done for 2 minutes, for example, or for 1 minute? No problem, because as you can see, the signal to noise ratio is pretty good here. The signal enhancement is very nice, so we can just decrease the integration time and obtain similar map with slightly higher signal to noise ratio for much faster than 4 minutes. But actually speed is usually not the aim of the tip enhanced Raman. The aim is the high sensitivity and high lateral resolution. So let's look at this data. Again, this is the corresponding intensity map of the Raman band of BCB molecules. This is the line cross-section, where we can estimate the resolution to be about 20 nanometers, and these are corresponding TERS spectra at different areas where there are molecules and where there are no molecules. Again, the overall enhancement of the tip without any recalculations is more than 100. Time for mapping was 4 minutes, and this is definitely not the fastest possible time. There was no problem to increase it, and the spatial resolution is of the order of 20 nanometers. Now, how about carbon nanotubes? This has always been a very nice sample to test different TERS configurations, TERS tips, TERS methodology, because this is a one-dimensional material. It gives a good, usually resonant Raman. It's a small object, so it's really a perfect thing to test your Raman instrument. And here I just collected some of the data, not all. I didn't have enough space on this screen to show all the data, but just some historical overview how NT-MDT instrument was used for successful TERS mapping of carbon nanotubes. In 2007 we delivered the data with about 50 nanometer resolution, and some of similar data was not published at a similar time. Now in 2010 and later on, our customers already successfully publish with a resolution 14, 20, 30 nanometers. So this is kind of a routine thing, 10 nanometer scale resolution imaging on carbon nanotubes. And carbon nanotubes still remain very attractive for testing, because again, this is a nice test to check whether your tip is working, your instrument is working, and how about optimizing the configuration of the TERS experiment. And now I'd like to show you some more intriguing data. This is again carbon nanotubes. This AFM topography, Raman D band, Raman G band. It's interesting to see that in some areas we don't actually observe nanotubes in topography, but we very brightly observe them on the Raman maps. This is corresponding overlay of G and D band. And now let us zoom in into this area and see what is there. And here it is. This is a many pixel tip-enhanced Raman spectrum of a carbon nanotube, overlay of G and D bands and with corresponding point spectra, and with a little bit of a caution, but taking cross section at the thinnest area at the end of the nanotube, we can probably claim coming to lateral resolution less, close to or less than 10 nanometers. That's good enough, but what is even more interesting, that this data was obtained not with a tuning fork probe, not with an STM probe, but with a simple commercial silicon cantilever, which is in mass production. We just applied to this cantilever NT-MDT proprietary coating and a proprietary TERS imaging mode. So from now on, with an NT-MDT instrument, AFM-Raman instrument, it's possible to deliver super sensitive, super high resolution tip-enhanced Raman with standard commercial silicon probes, which are and plus coating, which are easy to install, which are easy to use, which can operate in contact and non-contact regimes, and actually these are cantilevers for the mass production. And I invite you to stay with us, because another set of very impressive TERS data is yet to come. To finalize with carbon materials, I'll show you some more data on this is graphene oxide. You can see a high resolution TERS map corresponding point spectra and some magnified part of the sample from which we can estimate the resolution of the imaging to be close to 30 nanometers. That was done with a tuning fork AFM with etched silver wire. Graphene has been very successfully studied by the group of Renato Zenobi and ETH Zurich. This is the data and corresponding publication. It's a high resolution TERS map of CVD grown graphene on a copper substrate, and here you can see the 2D band intensity by green color and CH bands intensity by blue color. So in fact, instead of having a uniform monolayer, and this is actually a monolayer, so instead of having it uniform, it's possible to recognize by tip-enhanced Raman that there are different domains, the domains of pristine graphene and domains of hydrogen terminated graphene. As to mechanically exfoliated graphene on the right hand slide, it's a tip-enhanced Raman map of mechanically exfoliated graphene on a gold substrate, and here we can observe strong D band intensity at certain areas. So these are the areas of localized defects within the graphene monolayer flake. All this data was obtained with a resolution close to 10 nanometers and was done in so-called gap mode regime, where we have the gold surface sample and a TERS active STM tip on the top. Here is another example of TERS mapping of weakly sketching biological object. These are amyloid peptide nanotapes. This is STM topography and high resolution TERS map, where aromatic ring breathing band from a Raman fingerprint region is used for imaging. The major thing in this measurement is the sensitivity of the TERS technique, allowing us to see very weakly sketching objects. The resolution itself is also substantially beyond diffraction limit and is about 80 nanometers. Another very weakly scattering sample thiol monolayers. With the tip enhancement, it becomes possible to achieve considerable Raman signal with different signature Raman bands from a monolayer for a reasonable time, and apart from that it's possible to do two-dimensional mapping, so these are corresponding TERS maps of a patterned sample with two isometric thiols. The resolution power of TERS was not completely exploited in this experiment. Rather, the sensitivity and the enhancement of the tip-enhanced Raman spectroscopy technique was fully used to detect monolayer signal and to produce two-dimensional maps. Here is an example of how enhancement on the end of the TERS tip can be used not only for imaging but for modifying the sample. This is a thin layer of organic molecules on a gold substrate, more or less uniform layer. So this area was photo bleached by a TERS tip with a higher power laser focus to the end of the tip, so with a high laser power concentrated at the end of the nano antenna. And then the larger area was imaged with the same TERS tip. The Raman map was obtained with already a smaller non-damaging laser power, and we can see this is the photo bleached area. If we look at the cross section of the image, we get roughly about 75 nanometer step edge, and this step edge comes from two consecutive processes: first lithography and then imaging. So it means that each of the processes, lithography process and imaging process, produced the lateral resolution of about 50 nanometers. TERS mapping with NT-MDT instrument has also been successfully applied for semiconductor structures. Here is the periodically patterned silicon germanium layer on the silicon substrate, and at different areas with different thickness, the crystal lattice of silicon germanium is supposed to have different stress state. The period of the structure is well below the diffraction limit. Nevertheless, with the tip-enhanced Raman spectroscopy imaging, here is a Raman of silicon intensity band and Raman of silicon germanium intensity band and corresponding topography image, so the structure can be easily resolved, and the resolution estimation is about 50 nanometers. Moreover, point spectroscopy at different areas of the layer, at the thick area of the layer and in the thin area, show different Raman positions of silicon germanium band, and that corresponds to different stress state of crystal lattice. And it is my pleasure to show here the latest data done with NT-MDT instrument. This is the TERS mapping of individual double-strand DNA molecules. This was done by Dr. Evelina Lippitz and her colleagues in Australia using NT-MDT instrument in the group of Professor Don McNaughton and Baden Wood. So here you can see the AFM topography of the DNA structure on the microsurface. The AFM image is not specifically good, and that's mainly because the real TERS tip was used for this imaging, and this tip has a relatively thick silver coating, which is not particularly stable, and also the tip is not very sharp. Nevertheless, it's possible to locate on the AFM image individual double-strand DNA molecule, to see it on AFM height, on the AFM phase image, and to see the simultaneously measured TERS map of selected Raman region. And you can see the remarkable correlation of AFM map and the TERS map. That shows the reproducibility of the technique, and this is probably one of the first real two-dimensional images of DNA molecule, TERS images of DNA molecule well correlated with AFM topography. The resolution of TERS imaging from this data can be estimated to be of the order of 15 nanometers. But the major thing again about this experiment is not only the resolution but the sensitivity of the technique. The DNA molecules studied in this work were previously damaged by ultraviolet radiation, and actually the main target of the investigation was to identify double-strand breaks in DNA molecules induced by ultraviolet, and by statistical analysis of TERS spectra obtained from different molecules in different regions, it was possible to find 3 specific Raman signature spectra corresponding to 3 different types of double-strand breaks in individual DNA molecule, the breaks that were induced by ultraviolet. And in this slide I would like to summarize all the data generated using NT-MDT instrument by our own group and by our customers. If it comes to various TERS tips, then gold, silver and aluminum etched wires are used with our instrument to generate tip-enhanced Raman maps. Gold or silver coated silicon, silicon nitride, silicon oxide cantilevers, smart pre-calculated FIB processed probes are also being used. Regarding AFM feedback regimes, that is contact, non-contact with standard cantilevers, STM tunneling current feedback regime, and tuning fork with shear force and normal force feedback. Illumination and detection geometries being used are different: top illumination, top collection, bottom illumination, bottom collection, side illumination, and various configurations. Different lasers are applied with aluminum etched wires. People are using 355 nanometer laser, green laser for silver tips and for gold tips, blue laser and red laser for different tip materials, and most interestingly, different samples. TERS mapping was successfully done on carbon nanotubes and different nanowires, graphene and graphene oxide, silicon, silicon germanium, semiconductor nanostructures, polymers, thin organic layers of various types, various biological objects, amyloid fibrils, peptide nanotapes, lipid domains, and finally the individual DNA molecules. And in most of these experiments, people were aiming not just for single point spectroscopy but for high resolution 2D TERS mapping. I have demonstrated some of the data, not all of it because there is not enough time for that, but I tried to select the most representative TERS data done with NT-MDT instrument on various types of the samples. If you have not seen some data on some samples, please send me an email and I will be happy to send you the data or the publications. So now about TERS tips from NT-MDT. We are not only producing and installing the unique AFM-Raman-TERS equipment, we also did a big job on the TERS tip manufacturing technology, and now we can offer to NT-MDT AFM-Raman customers various TERS tips. That is etched TERS tips for STM regime, that is etched TERS tips attached to tuning fork for normal force and shear force AFM regimes. This is the etching device and protocols to produce TERS tips directly in the lab without need to buy it from us. And finally, and most interestingly, these are silicon cantilevers with NT-MDT proprietary TERS coating and with proprietary TERS imaging regime. And this cantilever probes are produced using standard cantilever manufacturing technology plus special coating, and that gives the really unique properties. These cantilevers are reproducible, reliable, they can be done in mass production, and they are very high quality. They are sharp, they have high resonance, high quality resonance curves. They can be done of various types, various frequencies, and so on and so forth. So this is a real mass production technology, not something which is handmade. So I would like to finalize my today's presentation with a scanning near field optical microscopy technique. This is another approach to optical imaging with a resolution beyond the diffraction limit. It works nicely with highly optically active samples which have a lot of light on the surface. It does not work with Raman, and this is a classical technique already, but still there are a lot of new interesting things appearing with applications, different probes and methodology. So first of all, NT-MDT is producing all types of SNOM probes. These are quartz SNOM fibers, etched fibers with a small aperture in the metal coating at the end that produces sub-diffraction limited resolution optical imaging. We produce such quartz fibers for different wavelength ranges, started from 400 nanometers and finishing with almost 1.8 micrometers. They're successfully used in all this wavelength range. We do tip and sample scanning in this SNOM configuration, and tip scanning is important for many of plasmonics and other applications when you don't want to move the sample. We are also manufacturing equipment and the probes for cantilever based scanning near field optical microscopy, and we manufacture both contact and non-contact probes, and we apply different measuring techniques with the non-contact probes. So here you can see just the basics of the SNOM operation: fiber SNOM in the collection regime and cantilever SNOM in transmission regime to measure optical properties of the surface, transmission or emission from the surface with sub-diffraction limited resolution. Again, I would like to repeat that this technique is not very sensitive and it would not work with Raman, but it would work with strong fluorescence or with other samples which have high concentration of electromagnetic field on the surface. So if you remember about the hybrid mode, just to repeat with this video, in a hybrid regime, but now let's take SNOM cantilever. So we are repeatedly touching the surface by the SNOM probe, moving up and down with a frequency about 2000 approach retract cycles per second, and at each approach and retract cycle we measure force distance curves. So we know about the sample interaction with the cantilever, and if we will simultaneously measure the optical signal, and this is what we're actually doing, so let's stop at this point. So if we measure a force distance curve and we measure simultaneously the optical signal and we record all this data, then we are getting the unique insight of tip sample mechanical and optical interaction, because we can correlate the optical signal with actual tip sample distance and with actual tip sample mechanical interaction, and by analyzing these curves, which are first pre-recorded and then can be processed, and by analyzing these curves we are able to extract a real near field signal with respect to the far field signal, and to increase the sensitivity and resolution of the technique. So this is integration of scanning near field optical microscopy with a hybrid regime. This is what we how we measure it, and we also can set up the maximum force acting between the sample and the cantilever, and that allows us to do careful examination of the sample without damaging the sensitive SNOM probe. And this is the example of test grading the hybrid SNOM imaging. So we move tip up and down again and record the complete optics versus distance curve, and then we can generate from this full three-dimensional data different slices. So this is actual SNOM signal, near field signal when the tip is in the contact with the sample, the red part of the force distance curve. This is image at about 100 nanometers from the surface. This is image 220 nanometers from the surface, and this image at 290 nanometers from the surface. And we can see that here we can see a real near field optical microscope image, and here we can start to see some diffraction patterns because the cantilever is not really measuring in the near field. This is a photonic crystal optical fiber. On the fiber cross section we can see the periodic whole structure that forms the photonic crystal, and SNOM was used to analyze how the light propagating inside the fiber is distributed in this photonic crystal. So SNOM probe was positioned on the surface of the fiber cross section, and it was scanned to collect the electromagnetic field localized in the fiber photonic crystal structure. And here with the red we can see the topography of the fiber surface, we can see the periodic whole structure of the photonic crystal, and with the green we can see light intensity, light distribution, and we can actually observe a perfect localization of light in the very center of the photonic crystal. And we can see the two lobes, which presumably comes from the second eigenmode of light in this photonic crystal structure. This is three-dimensional distribution of light emitted from the surface of semiconductor laser. This is the actual near-field pattern taken from exactly from the laser surface when the SNOM probe was very close to the surface. This is XY scan of the laser emission pattern, and this is the XZ scan, which actually shows us how the near-field emission pattern on the laser surface is gradually transformed into the far-field emission and forms the emission pattern of this certain laser. And the calculated XZ emission cross-section and the experimental one correspond with each other very well. Actually, these cross-sections were generated from a complete three-dimensional data that was first measured. This is the overall 3D intensity map of light on the top of the semiconductor laser, and the data I showed on the previous slides was just cross sections of this overall three-dimensional data cube. This is interference of propagating surface plasmons. In a gold film you can see the calculated structure of interference of different orthogonally propagating surface plasmons, and this is corresponding experimental result done by scanning near-field optical microscope in a collection regime. SNOM probe is scanning across the surface of the sample and is collecting by its aperture the intensity of localized electromagnetic field on the sample surface. Here SNOM is used to study whispering gallery modes in semiconductor microdisks. This is the microdisk topography, and this is corresponding distribution of light of two different whispering gallery modes. We can see the spectra of these modes and the corresponding light distribution across the sample. This was also done by SNOM in a collection regime. This is a sketching SNOM technique, so if we want to study intensity of localized electromagnetic field on the sample surface, we don't have to use aperture. We can just take a metal probe, and this metal probe will locally scatter the light by the very end of the probe, and the near field light will propagate into the far field being scattered by the tip, and if we detect this scattered light, we will be able to obtain distribution of localized electromagnetic radiation on the sample surface with very high resolution. This is the basics of sketching SNOM technique, and here is corresponding experimental data. This is an AFM image of a plasmonic lens, and this is corresponding calculated intensity of longitudinal component of electrical field of light in the very center of the disk, and this is corresponding sketching SNOM image, and the experimental data corresponds very well to the calculation. So in sketching SNOM it is possible to do high resolution mapping of light on a sample surface, and it's my pleasure to show here this data coming from the group of Andrei Kabashin in Marseille, and here they use sketching SNOM technique to detect localized electromagnetic field radiation in source substrates. Here you can see light localized at the very end of the individual gold nanoparticle, and when they change the light polarization, they can see the corresponding rotation of the light pattern on the nanoparticles. If we look at the scale bar, we can see that the resolution of this data is actually of the order of 10 nanometers, and this has been done again with scattering with apertureless SNOM by utilizing a special detection technique. At this point, I would like to finalize my talk. Here are some conclusions. I would like to emphasize again that all the data I demonstrated was generated using NT-MDT instrument, and this is actually a very small portion of the overall data and applications that we have. You can find more information on our website, and in the next couple of weeks we are going to put much more new data to the website, so please come to visit us again for the new information. Thanks for staying with us for so long time. I'm ready to answer your questions.